Hi, everybody. Thank you for coming to my talk today. And so I'm not initially sure about the audience and size of the talk and the content. So my talk is a very high-level overview about the phase 1 oncology trials. And how many of you work in oncology? Most of you, okay? So if you find it too easy, you can treat this as kind of an extended break. So as you mostly have known, the oncology is a special therapeutic area where those finding studies were not conducted in healthy volunteers. It's actually in cancer patients. So it's either in a variety of solid tumor patients or a variety of heme malignancies. So most of the time, they have been through multiple rounds of treatments until they get into the phase 1 domain. It's kind of they treat it as a last resort. So the goal of the study is to find the maximum tolerated dose, or MTD, using a series of small cohorts, usually 3 to 6 patients. And we will observe dose-limiting toxicity events, and these are usually pre-specified in the study protocols. And at the end of a phase 1 study, combining clinical safety, PK, and PD information, we will find both an MTD and recommended phase 2 dose, and these two may not be the same. So a recommended phase 2 dose may be slightly different from the maximum tolerated dose. So I'm going to first talk about the single-agent dose-finding study designs, and I will also touch briefly on the combination designs, which is the novel combination. My last slide is about combining safety and efficacy. I just want to briefly mention it so that I just want to raise awareness so that we in industry can use it more and more often, because in academia, in cancer centers, et cetera, they have been using this kind of design already. So a 3+3 design, as you've known, is that it's a traditional design that it's still prevalent in oncology studies and in companies. It's a very simple design where you enroll 3 patients and you see how many DLTs you have. It's either 0, 1, or 2, or 3. And you make decisions accordingly. If you see 1, you go up. If you see 0, you go up. If you see 1, you stay, enroll another 3. If you see 2, then you stop. And then if you enroll another 3 patients, you observe DLT rates and move accordingly. So as you know, that there are a lot of limitations about 3+3 design. It's very deterministic. It ignores the escalation history. For example, we as statisticians, we know that a bunch of 0 out of 3 DLTs, then you have a 2 out of 6 DLT, is different from just 1 0 out of 3 DLTs, and then you see 2 out of 3. But 3+3 ignores all the previous histories of 0 of 3 DLTs. And also, it also takes a same action under different kind of situations. For example, a 0 over 3 and a 1 over 6 leads to the same action, etc. And also with 3+3, a big drawback is that you cannot, once you go down, you cannot re-escalate. But with later model-based designs, if you go down, but if you later observe good data, you will be able to re-escalate. And also with 3+3, since it's deterministic, you don't have the flexibility on the cohort sizes. For example, you can't have a cohort of 4, etc. And also the performance of 3+3, as I mentioned, was already published elsewhere. So I don't have to belabor on this. And also Lee, the first speaker, has been mentioning about this. So she really laid out the ground for me, so I feel like I'm repeating everything from her. Another model-based design that was initially proposed, I think in the 1990s, was the continual reassessment method. It's a 1-parameter model-based design, really, where you can see in the formula, tau is the fixed parameter and theta is kind of having a beta distribution. And once you have the prior information and you have the model laid out in the bottom, then you can estimate, you can update the model and estimate the posterior DLT rates and make a decision accordingly. So this has been used, I think, pretty widely, and it's pretty well accepted too. And it's been shown that its performance is better than 3+3. So with that, I think another kind of an extension of the CRM design is the Bayesian logistic regression model, where Lee has also mentioned. So the Bayesian logistic regression model it is also a model-based design, and similar to CRM, we don't want to treat it as a model to the clinical team. So typically, what we as statisticians, we should say is that these model-based designs, because clinicians and other people, they will feel like you have a black box model and you come up with your recommendation and we as a team have to take it. So this slide was really, I wanted to show you that we wanted to present the model-based designs as a way that we have the data, we have the prior information, we have the trial information, the data we observe, we have the model there, and then we have estimated posterior, and then we recommend a dose. And once we have the recommended dose from a model-based design, we wanted to say that here is what the model recommends, but the real next dose decision is not just based on the model. A model recommends a dose, and then the clinical PK and PD information can be also discussed so that a decision can be made for the escalation decision. And this is really helpful in pushing the model-based design to study teams. So the model is really a two-parameter Bayesian logistic regression model, where the alpha and beta are assumed to have a very normal distribution. And the prior development is kind of an important piece as well. And then, but I think the publication has already laid out the methods for the prior development, so I don't want to belabor on that either. So the idea is that you enroll a cohort of flexible size X patients, usually 3 to 6, and you, similar to CRM, you update the model and estimate the posterior DLT rate for each of the dose level. And you can do escalation with overdose control as well, as Lee mentioned. And the cutoff really, people use different cutoffs for the overdose control. I think Novartis has been using this model for about 10 years or maybe over 10 years. They use 25%. In some other places, as I've seen in past JSM talks, for example, in Onyx, they use, I think, 35%-ish. So it really depends on your tolerance on the overdose and the drug profile. And also, the recommended dose will be the dose with the highest posterior probability in the target DLT rate level, given that the overdose criteria is satisfied. As you have seen, that basically you have different doses, and you will choose the dose that is not red, but the highest here that is in the target level. And these graphs help understanding the design and the decision. So another advantage about the Bayesian logistic regression model is that not only can it be flexible because it had two parameters compared to the CRM models, and also we've done simulations to show that its performance is also superior than the CRM design, because the dose DLT relationship, if you kind of plot out the relationship, it's more flexible. You have different kind of beta shapes compared to the CRM, where in the middle it's kind of more parallel in the space. So an advantage of BLRM is that you can add more covariates to the model, as you can see here. So the other parts are the same, the prior and everything, but if you add a prior, you can incorporate the different characteristics of your study. For example, if you have run a dose escalation trial with drugs with different formulation, you can parameterize the formulation of the drug into your model here. Or if you are trying different dosing schedules, in your dose escalation study, you can also try to formally incorporate that here. Another scenario you can use the BLRM with covariates is when you have combination studies, but not entirely novel combinations where you have no idea where the two doses are. If you have a combination of one new agent, another is a more established drug, but the established drug you want to try out maybe 2 or 3 different doses instead of fix the dose at the marketed level. So in that case, you can try to use the dose of the other established drug as a covariate here, like one. So here is an example of the single agent BLRM with covariates where you have a couple different doses, and this is formula simulation. So these are the assumed prior DLT rates for assuming you have 2 different formulations where prior to the study you know that one of them is a little bit more toxic compared to the other. So if you use a model with covariates, then if you use the model with covariates, then the decision to escalate or to not escalate would be different. Here is the probability of overdose. So here for the 2 different formulations, you will have 2 different you will have 2 different overdose control probabilities. So if you have one of the formulations, you won't be able to use this dose. And if you have the other formulation, you'll be able to use it. So another design for single agent study is called modified toxicity probability interval. So we internally have not used it, but we are trying to start evaluating and use it. So it's a design also in a Bayesian framework with a beta binomial model to compute. It's a probability of 3 intervals similar to the BLRM. They have also 3 intervals. So that has the relative distance between the toxicity rate of each dose level to the target probability. So they have 3 underdosing interval, and they have proper dosing interval, they have overdosing interval. So I think the beauty of the MTPI method is that it's a really, because the model-based designs is more like a black box to the team, as I mentioned before. And before the study start, you don't really have a good idea about what's going to happen during the course of the study. 3+3 clinicians like it because it's very easy, and they know what to do once they see the number of DLTs, et cetera. And MTPI is easy because based on your calculations prior to the study, you can come up with such a table. It's like a decision table where you will know for number of patients treated at a particular dose, and for a particular toxicity, you will know whether to escalate or to stay or de-escalate or discontinue. So the decision is very easy so that nobody is confused and nobody feel like they don't know anything. So this is that's the benefit of this design. And also, there have been simulations as well to compare MTPI to CRM to 3+3. And I think MTPI is also superior to CRM. So next, I'm going to move to the combination study designs, where you can run kind of novel combination studies. For example, I'm not sure about the biological reasons, but for example, if you run a combination with a MEK inhibitor plus a RAF inhibitor, and they are both new molecular entities, they have not been approved, but usually when you are at the point of running the novel combination, you will have already run the single agent first in human study, so you have kind of a little information about the toxicity of the single agent study. And so there are different ways you can run the novel combinations. And what I'm talking about today is a very partial list of the things that you can do. So one thing you can do is called 3+3+3. It's kind of like an extension of 3+3, where you now have 2 agents, as you can see in the graph, and you can make decisions about where to escalate based on what you observe what you observe in your data. I'm not going to talk about 3+3+3 in detail. I will just briefly mention about the Bayesian logistic regression model for combination dose findings. So with a slight reparameterization of the original BLRM for combination models, as you can see, it's very easily interpretable, where you can see the marginal DLT rates are parameterized as exactly the same as the single agent models. And also you have a parameter about the interaction of the two drugs. So with this interaction, you can use different kind of priors for the interactions, because when you run the combinations, they can either have synergistic effect or antagonistic effect, or they don't have much interaction. So you can incorporate that information. Also, typically, you can have this information from preclinical experiments about their toxicity interaction. And you can either put in normal or log normal distribution for the prior for the eta. And also, you can use just the priors for the single agent models. And they could be by very normal. And once you've run the single agent studies, you already had some data, and you can use the kind of the posterior distribution from the single agent studies to start as the prior for the combination. So for a combination study, it's a little bit more complex. And the goal of the combination study is to find maximum tolerated dose or recommended phase 2 dose. For a single agent study, it's easier because you have a one-dimensional, one dimension of dose where you can identify an MTD. But for combination study, it becomes a matrix, and then you will sometimes be able to find multiple MTDs, because that's just the way it is. And when you try to find MTD, sometimes we've run simulations where when you try to find your MTD and you, just by chance, sometimes you can wander off in the matrix and go somewhere that you are stuck there. You can't really get out of that area to go to the true MTD area. So a hypothetical study could be that you evaluate DLTs in the first cycle, and then you have some exposure requirements for the different drugs, because they have to take at least a certain amount to be valuable. And the toxicity interval can be a little bit more relaxed compared to single agent. A single agent is 0.16 to 0.33. And also you can use the escalation with overdose control. And before we kind of tried escalation, we call it, one of them is fast escalation, where you can escalate both. And you can try the slow escalation, where you can only escalate one. But I think there could be regulatory concerns about doing escalations for both drugs at the same time, because if there are things happening, you don't really know what's contributing to it. But with a slow escalation, where you escalate one at a time, it's easier that you figure out what went wrong if something happens. And also, you can't escalate over 100% from the current dose level. So if you, this is a Novartis slide actually from my material. So if you run something like that, you can calculate the posterior probability of the DLT, and also you can see that if you cross out those red that didn't satisfy the overdose controls, you can also see that you can't escalate at the same time. Then you can figure out, if you cross out these and you know where you are before, you can easily see there could be multiple cells that you can go to from where you are. Say you are here, for example. And the choice about where to go next, you can open up, for example, 2 or 3 parallel cohorts to explore so that your trial runs faster. Or you can also choose to run one of them to see what happens. and then enroll the next dose not in parallel, but sequentially, so that you can take more time to run the trial, but it's more safe. So it's really a plot that gives you the choices of your next dose, and you can discuss with your team about what to do next. So this is the combination of safety and efficacy information in those finding studies, and these are more for single agent. So there are various publications about the combining safety and efficacy information designs, and most of them are Bayesian designs. For example, Bayesian dose finding in 2 treatment cycles based on the joint utility of efficacy and safety, and also the team's design. And I just want to briefly mention the last one. Just the idea of it is the Bayesian adaptive dose finding based on efficacy and toxicity. It's a review paper by Peter Thaw, where one of the design he mentioned is the utility function, where right now we are running our phase 1 designs looking only at the toxicity. We are pretty much ignoring the safety information. Once we find the MTD, we open up cohorts. Maybe a couple of cohorts to see, or at the MTD cohort, we start seeing the efficacy information. But so for this kind of design, you jointly model the toxicity and efficacy information, and you discuss with your clinician about the scores, the utility scores that you can see. For example, so it's kind of like you have a trade-off between toxicity and safety. If you can have a CR, maybe you can tolerate more toxicity, right? A CR with high severity toxicity is a score of 75. But if you have a PD, you will have much less tolerance of toxicity because you're not seeing the toxicity effect. Why do you have to endure that toxicity? So I think all of these kind of new designs are really good for us to study and maybe try to explore in our studies. I know there are regulatory concerns and other things that we have to pass the hurdle, but I think it's a really good way for us to experiment around these new designs. And also for Cytel, if you can implement these new designs for us to use, we'll be very happily try them, because if you implemented them, it's much easier for us to use them and then not having to spend a very long time and having to implement them. That's my last slide.